morning. This lecture video is going to go over the electrical activity of the heart, including a description of what is going on or how to read an ECG. Let's begin with the idea of autorhythmicity. The heart is unique in that it can initiate its own action potential, and it can do so at a fixed rate, which then is going to trigger our heart's ability to contract. This is very unique to cardiac muscle, so smooth muscle cannot do that. It has to get its action potential stimulated by an outside force, and same with skeletal muscles. We need to have those neurons cause contraction to occur. So a heart is unique in that it can generate its own action potential and thus manage its own contraction. The heart activity is still influenced by the nervous system and the endocrine system depending on what's happening, but its mechanism is self-generated. Now, there are two types of cells that are found in cardiac muscle. Myocardial contractile cells, and this is the vast majority of the cells. There's 99% of the cells that are in the atria and the ventricles are going to be the contractile cells, and their job is to conduct impulses that cause contractions, which ultimately is going to pump blood through the body. There are also myocardial conductive cells or conduction cells, and these are going to be smaller, and there's fewer of them. They make up only about 1% of the total cells in the atrium and ventricles, and their job is to basically start and spread action potentials that then travel throughout the heart and trigger contractions. So two different types of cells. Myocardial conduction cells work very much like a neuron. So we're going to talk later in this lecture video about depolarization, repolarization, and that whole process of generating an action potential that we did way back in the nervous system. Same general processes with just a little different tweaks due to the fact that we're dealing with cardiac muscle. But conduction cells are going to be very much like neurons. If we are looking at cardiac muscle and how it is arranged in comparison, particularly to skeletal muscle, because we talked about this stuff already when we went into skeletal muscle contractions and sarcomeres and Z-line to Z-line and all of the activities in the sliding filament theory, right? Well, cardiac muscle is going to work in very much the same way. In general, a sarcomere of a cardiac muscle is the same as a skeletal muscle. You still got the thin, thick filaments. The sliding filament theory still is going to occur, pulling those filaments towards the center of a sarcomere, shortening it. A couple differences: cardiac muscle fibers are typically shorter than skeletal muscle fibers. They have a little bit smaller diameter to them, so size-wise, they're not quite as large or as long as a skeletal muscle fiber. They are striated, just like the skeletal muscle fibers. So remember, those were the A bands and the I bands. And another chemical difference is that the sarcoplasmic reticulum, so the endoplasmic reticulum of a muscle cell, is going to hold fewer calcium ions than in a skeletal muscle. And this is going to be important when we talk about the action potential and what's happening, because it's kind of going to cause the contraction to be a little bit slower to start in a cardiac muscle than in a skeletal muscle. Cardiac muscles have a ton of mitochondria, which would make sense because it's continuously generating contractions, right? So we need to make sure that we have enough mitochondria to make the energy that it needs. Cardiac muscles also have a special structure in them called intercalated discs. These are the connections between one cardiac muscle cell and another cardiac muscle cell. And these are going to be really important because they help communicate with the cells from one cell to another, and this is going to allow muscles to contract in a very synchronized way. So if you see these line patterns here, and I'll show you an actual micrograph in a second, these are the intercalated discs. So really, they play an important role in cell-to-cell communication. Cardiac muscles typically are going to use aerobic respiration, meaning that they're going to metabolize lipids and carbs for their energy sources. And we're going to talk a little bit more about the action potential in a second, but you can see that there are some differences in how the action potential works compared to that of a cardiac or of a skeletal muscle. Here's a tissue micrograph of cardiac muscles. Again, usually they only have one cell per muscle. One nucleus per cell. So you can kind of see that here. Every once in a while there might be two, but in general it's uninuclear. You can see these lines here, these dark lines, those are going to be the intercalated discs, and you can still see here that it's striated, just like skeletal muscles. So there's an intercalated disc, there's an intercalated disc, nucleus. So that's kind of how skeletal tissue ends up being, cardiac tissue ends up being arranged. So let's look at what's going to happen here with the electrical signal as it travels its way down into or through the heart. There are a couple different parts or groupings of electrical cells that are found within the heart. And there's a diagram that is posted. You can print that out and use that to help you write these in if that's helpful for you. But the things that we're going to be looking at are the SA node, which is going to be located right here. Then we're going to be looking at the AV node, or the atrioventricular node, which is right here. The atrioventricular bundles, which are going to be right here. And then there's going to be a right branch and a left branch. And then the Purkinje fibers, which are going to come off of these branches. So those are kind of the structures that we're going to be looking at that are going to be responsible for passing the electrical signal through the heart, allowing the heart to contract, specifically allowing it to contract atriums first, ventricles next, atriums first, ventricles next, and pushing blood throughout the system. So we're going to start with the SA node. The SA node is called the pacemaker, okay? It is going to be responsible for setting the pace of the heart. It is located in the right arterial wall, and you can see where it is right here. It generates an impulse of about 75 to 80 times per minute. So that's where you're getting your heartbeats per minute at. It spontaneously generates the action potentials. We sometimes call this the sinus rhythm in terms of what it actually creates when we're looking at the different electrical activities. And its job is going to be to send the activity or send the impulse through a pathway called the internodal pathways to the AV node, which is located right here, right above that tricuspid valve between the right atrium and the right ventricle. The time that it takes for the SA node to send its signal through those internodal pathways to the AV node is going to take about 50 milliseconds. So once we travel through those pathways, then we are going to hit the electrical signal will hit the atrioventricular node. This is located again in the inferior part here of the septum, right above the tricuspid valve. When the AV node, at the AV node, the impulse coming from the SA node is actually going to be delayed for a second. And this is really important because this delay is going to allow then, once this is contracting, the atriums are contracting, to allow that atrium to basically push all of that blood to the ventricle and pausing it so that we're making sure that that contraction comes to completion and all of that blood is getting pushed through. It's a pretty slow impulse conductor, so that's why, in part because you have that pause there, and the reason is just, again, to make sure we're getting all of the blood out as we squeeze into the ventricle. The AV node is then going to send the signal to the next electrical pathway, which is going to be called the atrioventricular bundles. This is found in the superior part of the septum here. It's really the only electrical signal or connection between the atriums and the ventricles. And as the signal travels down through these AV bundles, it will eventually branch into bundle branches. One that will go around the left ventricle towards the apex, one that's going to go around the right ventricle towards the apex. So basically, we're going to be generating this contraction and the impulse needed for it all the way around each ventricle, so each ventricle can then contract. Because remember that the atriums contract at the same time, left and right, and the ventricles contract at the same time. We often call these bundles or these branch bundles the bundle of His. Coming off of these branch bundles, you can see they're kind of tiny on here, these little fibers called the Purkinje fibers, and their job is to, again, just kind of propagate this action potential. And what they're going to do is they're going to allow the ventricles to basically contract starting at the bottom, and the contraction will move itself up towards the base. And that's because of these Purkinje fibers. So ventricles almost contract in the opposite direction that you would think they would, right? So the atriums contract, it sends that signal down. That signal is going to cause this part of the ventricle to start contracting, and then at the end of the full contraction, the top part of the ventricle will be contracted. And that's because of these right bundle branches and these Purkinje fibers. So again, you can kind of see the AV bundle is going to conduct that impulse right through from the SA node to the AV node. It's going to travel on down through this septum. And once that branches and comes up, then the ventricles are going to contract, going from apex to base. So let's look at exactly how this works when we're talking about an action potential. So you need to kind of review in your own mind the steps of the action potential that we talked about in a neuron. So we talked about how in a cell, there is a difference in charge from the outside to the inside, and this is called being polarized. So in a typical cell, the outside is going to be more positive than the inside. So inside of cells, we have a whole bunch of potassium, and then on the outside, we have a whole bunch of sodiums and calciums, and those are going to cause an actual difference in charge between the outside and the inside. And this difference in charge and basically the reversal of this difference in charge is what's going to drive or move an electrical impulse along the cell. And just like in a neuron, as we travel down the length of the axon, this same process is going to occur in a muscle fiber. However, remember we have two different kinds of cells. We have these pacemaker cells, and we also have the contractile cells, and their action potentials are going to be slightly different. Still going to have some of the basic features. Depolarization is going to occur, the action potential, repolarization is going to occur, but there are some slight differences between that are important to at least discuss. So if you look at a pacemaker cells, unlike other cells, don't have a true resting potential. They're going to have, or a static one, they're going to have a resting potential at or around about -60 millivolts, and it's going to spontaneously generate or spontaneously move up to -40. And this is the threshold. And remember, the threshold is going to be that point where ion channels start to open up, and we are going to get the active process of depolarization or becoming more positive is going to occur. So in this action potential, we started at -60. We're going to spontaneously bump up to -40, which is the threshold. And then at that point, we're going to get into active depolarization. So when we hit that threshold, the calcium channels are going to open, and calcium is going to flow into the cell, and it's going to cause the insides to start to become more positive. So that's going to cause depolarization to occur, right? Like up here in this 0 red phase, because the calcium ions are bringing in a positive charge into the inside of the cell. And then at the very peak here of depolarization, what's going to happen is the potassium channels are going to open, the calcium channels are going to close. Potassium then is going to leave the cell and go back to where they came from, back to the outside of the cell, thus causing the inside of the cell to start to become more negative again, okay? And then this process is going to kind of start all over, but there's going to be this time period of hyperpolarization where the sodium is going to start to come back in, but it's going to do so very slowly again until it reaches the threshold, and then depolarization occurs and the process starts all over again, right? So just to review, at this point, sodium's coming into the cell. It does it very, very slowly in a cardiac pacemaker cell. Once we get to a point where we've become slightly more positive than where we started, I mean, we're only going from negative 60 millivolts to negative 40, then we're going to trigger all of those calcium and potassium gates to open. So potassium is going to come into the cell, influx, so depolarization is going to occur. Then we're going to, potassium is going to start to leave the cell, so potassium gates will get, or potassium will start to get pumped back out of the cell and get back down to where the in general resting potential is, even though we don't, it varies in a pacemaker cell. So you can kind of see that pattern for a pacemaker cell, very similar to a neuron. This process is very, very similar. Now, if we look at what's happening in a contractile cell, there are a couple of really important differences here. So again, contractile cells are going to have a different set of ion channels. So they still have the sodium, they still have the calcium, they still have the potassium ion channels. One thing that's different between contractile cells and pacemaker cells is these have a very consistent resting potential. So again, it's going to be at about negative 90 millivolts. So again, the resting potential is different in a contractile cell than it is in a regular neuron or in a pacemaker cell. It's going to depolarize or start this depolarization process only when stimulated, usually by neighboring myocytes. So again, this is where these intercalated discs come in. Once this occurs in one cell, it's going to pass this action potential on to the next cell. Once it gets to about negative 70, which is the threshold roughly, sodium channels are going to open and they're going to cause a rapid entry of sodium into the cell, and this causes a sharp rise in voltage. So that's going to be right here in blue, rapid depolarization. At the very peak then, the sodium channels are going to close, the potassium channels are going to open, and this is going to result in a small decrease then. You can see that right here in green because the potassium is leaving. A small decrease in membrane potential, which is kind of like early repolarization, for lack of a better term. What also happens is that calcium channels are going to kind of remain open, moving calcium into that cell membrane. And because you have this calcium that's still coming in, even though potassium is going out, it basically causes a balance. And we have this period of stability in charge. And it's just because, you know, the calcium and the potassium are kind of balancing each other out. This stage, which is called the plateau stage, this is unique to contractile cells and super important because this is kind of what's going to give our heart the ability to kind of pause to make sure that everything has been pumped out before it continues on with the contraction. So at this point then, at some point, we're going to end up going into this, what's labeled as phase 3 on here, where the calcium channels are going to slowly close off. We still have potassium channels that are open and spitting the potassium back out to the outside of the cell, and that's going to cause this rapid repolarization to occur. And that's going to bring that membrane potential back down. At the same time, calcium ions are going to be pumped out of the cell. The sodium and potassium pump are going to become activated to make sure that we are bringing the membrane potential back to where we started. And that is going to be at a roughly negative 90 millivolts. So because of this plateau phase, which again is unique to this electrical activity of cells, the cardiac muscle can stay contracted for a little bit longer period than a skeletal muscle can. And again, this is going to allow the blood to be fully pumped out of the chambers before the next phase starts. So this unique plateau phase is unique. It's different than in skeletal muscles and in pacemaker muscle or pacemaker cells, but it's a really important phase because it's going to allow our blood to be pumped out of the different chambers. Now, we can look at these different types of electrical activity. We can take a monitor and we can monitor what's happening with these different millivolts and electrical activity and apply them then to the different types of activity that's occurring within the muscle. And this is called an electrocardiograph or an ECG. It's basically a composite of all of the action potentials generated at a given time. It's not a tracking of one action potential, however. So just know that it's kind of an overall picture of everything that's happening at one time. There are three distinct waves called deflection waves that we're going to talk about, and then segments between them that indicate things that are happening in the heart. So typically we refer to the P wave, the T wave, and then there's a QRS segment or QRS wave. But again, each one of these is representing a specific activity of the heart. So starting with the P wave, this first P wave lasts about 0.8 seconds, and it results from the movement of depolarization wave from the SA node to the atriums or through the atrium. It is going to, this is basically, in layman's term, this is depolarization of the atriums, okay? Now, the distance between P wave to Q wave is going to represent how long it's taking the AV, the signal from the AV node to get to the SA node through those interpolar nodules or pathways, internodule pathways. So then we get down to the Q wave. The Q wave is going to represent depolarization of the intraventricular septum. So as those swapping of charges occurs along the septum between the ventricles. And then we are going to get up to the R wave, going back up to the top. The R wave to the S wave represents depolarization of the main mass of the ventricles. So remember that that depolarization or that contraction is going to start at the apex, which represents the R wave, and it's going to move towards the base or the top of the heart, which kind of represents the S wave. So when we look at the QRS wave as a whole group, this is the electrical activity that's occurring as the ventricles depolarize. Now, because the atrium's depolarized first, right, they are going to be going actively through repolarization at the exact same time that the ventricles are depolarizing. However, the electrical activity is hidden by the dominant ventricular depolarization. And so basically the ventricular contraction is going to show up more dominantly over the atriums basically relaxing, right? So as the ventricles contract, the atriums are relaxing. And as the atriums contract, the ventricles are going to be repolarizing. So kind of keep that in the back of your mind that this is occurring at the same time, even though we don't necessarily see it on the ECG, we just refer to this QRS wave as ventricular depolarization. So then we got the ST segment. Whoops. The ST segment, it refers to that plateau that we talked about earlier in the myocardial action potential when the ventricles are contracting and pumping that blood out of the body. And so then last is the T wave. The T wave represents ventricular repolarization immediately before relaxation, also called diastole, ventricular diastole. And then this is going to basically repeat itself, continuing the steady state of muscle contraction, atrium contraction, ventricle contraction, atrial contraction, ventricle contractions, all the way through or all the way every single day of our lives. Repolarization, as you can see, is a lot slower than the process of depolarization. And again, part of that is just making sure that we have the time needed to give the heart a moment to relax before it can start to fully contract again. Here's a diagram, again, showing some of these different things that are happening. So the SA node is generating an impulse, and that's where this is going to be coming in here. The atriums basically contract, and there's a delay that's going to occur before it hits the AV node. That's this part of the wave. It's going to then travel down through the bundle fibers to the apex of the heart or ventricles. It's located right here. These are then going to contract, depolarize, causing ventricular contraction, which again is this QRS wave. And then we're going to end up relaxing and starting this process all the way back over again. So this is when we look at an ECG, we're going to be measuring the waves, the distance between each of these waves, and corresponding them to a time, because we know that the heart does things at a pretty consistent time interval in order to look for abnormalities that might occur.